Let's move on to our second speaker today. It is my pleasure to introduce you to Dr. Jon Zugazagoitia from the Department of Pathology, Yale University School of Medicine. The title of his talk is Discovery of Biomarkers Associated with Benefit from PD-1 Checkpoint Blockade in Non-Small Cell Lung Cancer using High-plex digital spatial profiling. Please join me in welcoming Dr. Zugazagoitia. I will turn the presentation over to you.
Okay, good morning, everyone. And thank you, first of all, to NanoString and all the organizers for inviting me to share our work with you in this nice virtual conference. And this presentation will be related to a work that we did when I was at Yale related to discovery of new biomarkers of response to immune checkpoint therapy in non-small cell lung cancer using digital spatial profiling. So first of all, I'm going to start with an introduction, sharing with you the idea of why we need new biomarkers of response to immunotherapy, particularly in lung cancer, and what are the issues with some of the biomarkers that we are using currently in the clinic, such as PD-1. So as all of you know, most of the patients with lung cancer that receive PD-1 axis inhibitors actually do not benefit from them in the long term. As you can see on the left-hand side, the 5-year overall survival with nivolumab in this case is around 15% in unselected non-small cell lung cancer patients. If we select them for PD-1 status, meaning that if we give these drugs to patients with a PD-1 tumor proportion score greater than 50%, which are those more likely to benefit from these drugs, even in this ideal situation, not the majority of patients actually do not benefit from the drugs in the long term. As you can see, response rate are around 40% in this high PD-1 population, and the 5-year overall survival still remains to a minority of patients, about 30% of them. So this clearly shows us that we need better biomarkers, particularly for monotherapy or for selecting candidate patients for monotherapy with these drugs. And PD-1, as you know, is the only biomarker that we currently use in the clinic to select patients to give these drugs. And as you already know, I'm not going to spend too much time on this, but PD-1 has its limitations, and I'm going to share with you two of those that I think are more important. First of all, I'm showing here, this is the way pathologists score PD-1, and this is the way pathologists tell us the amount of PD-1 that there is in a given tumor of a patient. And as you can see here, PD-1 scoring by pathologists is inherently qualitative. They give us the proportion of cells, the proportion of tumor cells that are positive for PD-1, but this assay doesn't really take into account the staining, or at least the intensity of the staining. So it's actually a qualitative assay, and we are losing a lot of information here. And the other limitation that I see as an important limitation in the clinic is that although there is good concordance between pathologists to score tumor cell PD-1, as you can see here, the ICC scores for pathologist concordance are above 0.8, which is good for tumor cell scoring, but it's not good at all for immune cell scoring, as you can see here. And we don't really know if the predictive value of PD-1 comes from tumor cells or immune cells, and this is something important to take into account. And this poor intraobservable reproducibility for immune cell PD-1 scoring has been shown in other studies. This is the second one that came up to the same conclusion, that pathologists there is good concordance between pathologists for scoring tumor cell PD-1, but not so much for immune cell PD-1. So at the end of the day, particularly for immune cell PD-1, this is not a very reproducible assay, and we need more objective or automated methods for scoring PD-1 and also other markers. So with this idea, we kind of developed, or we initiated a study with the aim to identify biomarkers of response, in this case to single-agent PD-1 checkpoint blockade, using geomics digital spatial profiling. So what we did is we took a retrospective cohort of immunotherapy-treated non-small cell lung cancer patients at Yale at a single institution. We collected their tumors, and these tumors were represented in a tissue microarray. We assessed 2 non-adjacent tumor cores per non-small cell lung cancer case, using, as I said, the geomics beta system that we had at Yale a year ago or so. And we used the 44-plex protein panel, and these proteins were assessed simultaneously in 4 different tissue compartments, as we will see later. And for the statistical analysis, we did a compartment-specific outcome association analysis for progression-free survival, overall survival, and clinical benefit, exploring 2 different cut points for each marker. As you can see, we did multiple comparisons here, but well, this was initiated as a discovery study, so we are allowed to do multiple comparisons. We did actually multiplicity adjustment in order to avoid false discoveries, and we used the Benjamini-Hochberg false discovery adjustment method. And we adjusted for a number of comparisons per compartment and separately for each of the explored cut points. And finally, we did multivariate analysis in order to find markers that were significantly associated with outcome, independently from clinical prognostic factors, in this case, performance status, smoking history, presence of liver metastasis, or lipid score, which we know is strongly prognostic in these patients. So that slide, and well, this is the cohort that we used. I'm explaining here the clinical characteristics of this retrospective cohort. It contains a total of 81 cases, and these are the clinical characteristics here that resembles pretty much what we usually see in a routine clinical setting. As you can see, these were patients treated with a variety of different treatments, but we were particularly interested in this subgroup of patients receiving monotherapy with PD-1 or PD-L1 inhibitors. And this was the panel, the protein panel that we used in this case for discovery of biomarkers in spatial context. This has a total of 40 markers plus controls. And these are markers related, as you know, with immune cell composition, also markers related to functional orientation of the immune infiltration, and also with some targets that are important for T cells particularly. And we used 3 visualization markers, in this case antibodies to detect tumor cells with pancytokeratin, to detect immune cells with CD45, and to detect specifically macrophages with CD68 staining. So this is how the kind of images that we got in this study. This is showing a TME spot stained with 3 different visualization antibodies, as you can see here. In green, cytokeratin positive tumor cells. In yellow, CD45 positive immune cells. And in red, these are CD68 positive macrophages, as you can see. And in blue, these are cells that, as you can see, they are in the stroma as well, but do not stain positive either for CD45 or CD68. They are presumably fibroblasts. And this is the way that the geomic system actually does tissue compartmentalization. Remember that our primary objective here was to identify biomarkers associated with outcome in spatial context. So that's why we measured all these 40 markers in 4 different compartments. And this compartmentalization is shown here. the system kind of colocalizes the fluorescent signal to create this tissue compartment or masks, as you can see here. And this is the epithelial or tumor compartment shown in green, the leukocyte compartment shown in yellow, specifically the macrophage compartment shown in blue here, and finally a fourth compartment, which is the non-immune cell stroma, which are cells, as I said before, that are positive for nuclear staining, but they are negative for cytokeratin CD45 or CD68. And then, in order to make all the samples as comparable as possible, we did area and normalization. So first of all, samples or compartments with less than 10 nuclei or less than 100 micron surface area were excluded from the analysis. At least in our hands, it was very difficult to obtain reliable quantitative information for very small areas. Remember that we are measuring immune markers that sometimes they are expressed, or usually they are expressed at very low levels. So it was hard to quantify them from very, very tiny areas. That's why we kind of excluded them from this analysis. Those samples that passed this filter were then normalized by surface area, as I said, in order to make all the samples as comparable as possible. And we use this formula. And this is kind of summarizing all the methods that we used in this Consort diagram. So we had a total of 81 cases included in YTMA 404. From these cases, a total of 67 cases were finally collected from 2 different blocks. These blocks contain tumor cores that are non-adjacent from each of the cases. And as you can see here, from these 67 cases, 53 of those had predetermined biopsies and received single-agent PD-1 checkpoint blockade. And this was our target population. This was our true discovery cohort. So in these 53 cases, we measured 40 immune parameters in 4 different compartments: tumor compartment, CD45 compartment, CD68 compartment, and finally non-immune cell stroma. And these are the number of valuable cases per compartment after excluding those that had very small areas or areas with less than 10 nuclei. As you can see, for example, there were 42 evaluable patients for CD45-related discoveries, because not all tumors, as you know, contain immune infiltration, so we had to exclude them for these CD45-related discoveries. So I will start showing you some analytic results first. As you can see, first, in order to prove the performance of the assay, we kind of calculated the normalized counts for each of the markers, and then we also calculated the signal-to-background ratio in order to show the performance of the assay. I'm showing here an example of one of the markers that was showing really high counts and really good signal-to-background ratios. This was the case for STING measured in the tumor compartment. As you can see, a lot of tumors express high levels of STING, and most of the cases have signal-to-background ratios way above background. As you can see, way above 3 or way above 5. This is a marker that is showing really good performance. But there were also other markers that, as you can see here, they were expressed as at lower levels. So in this case, the signal-to-background ratios for some of these markers, in this case LAC3, was below 3, meaning that we cannot really distinguish between true signal from background. And this was the case from 5 markers, as you can see here, which had signal-to-background ratios less than 3 in all compartments and in the vast majority of the cases. So we decided to exclude these markers from outcome analysis because we weren't really, or we couldn't really distinguish true signal from background from these markers. And also another thing that we realized was that the 4th compartment, which was the non-immune cell stromal compartment, in this compartment, there were really low counts for most of the immune markers, which is not surprising because we are measuring immune markers in a tissue compartment which doesn't have immune cells. So at the end of the day, this was also some kind of validation for us in order to prove that we were actually doing a spatial measurement. As you can see here, there was almost no CD3 counts in the non-immune cell stroma compartment as compared to the CD45 compartment. And also, we kind of explored what was the correlation between counts from different areas of the tumor per case. And as you can see, there were markers where the regression between different areas of the tumor was quite good, in the range of 0.6 and 0.7 in some of the cases. This was true for STING or HLA-DR. But there were other markers where the heterogeneity was substantially higher and the correlation between different areas of the tumor was lower. And actually, this was the case for those immune markers that are expressed at lower levels or they are more heterogeneous. So at the end, this regression is showing us, or is a function of marker abundance and tissue heterogeneity, as you can see here. And finally, we did the outcome analysis. This was the primary objective of the study. As you can see here, we found a number of markers that were associated with outcome in spatial context. Actually, there were 18 markers associated with either progression-free survival or overall survival, as you can see on the table in the left-hand side. Those were markers that were associated with outcome in the univariate analysis. After doing multiplicity adjustment and doing also multivariate analysis, there were 5 markers that remained significantly associated with outcome. And those 5 markers were VISTA and CD127, which is the receptor for IL-7 in the tumor compartment, and CD4, beta-2 microglobulin, and CD3 were markers associated with outcome in the CD45 compartment. There were also other markers that were significant in the univariate analysis, but they didn't hold significant, and after adjusting for multiplicity or after controlling for clinical factors. This was the case for PD-1, for example. PD-1 was associated with overall survival only when measured in the immune cells or specifically in macrophages. As you can see here, we didn't find significance for PD-1 when PD-1 was measured in the tumor compartment. And there was also other markers more classically associated with outcome as well, such as CD8 here, which was associated with overall survival, but again, it didn't hold significant after adjusting for multiple comparisons, kind of highlighting the idea that perhaps it's not all about CD8 cells. There are other cells and other markers that are contributing to the benefit of immune checkpoint blockade in lung cancer. And just to highlight the most relevant discoveries for us, I'm showing them here, that high levels of CD4 and CD56, which is a marker for natural killer cells in the CD45 compartment, both were predicted for all clinical outcomes, including response rate, longer progression-free survival, and longer overall survival. And this was true after controlling for clinical factors. And high levels of VISTA and CD127 in the tumor compartment were markers associated with resistance, actually shorter progression-free survival. This was also a remarkable finding because we really need markers with high negative predictive value in order to exclude those patients that are not going to benefit from these drugs. And this was a finding that we thought it was interesting. So I'm going to start showing the outcomes for PD-1. As you can see, PD-1 was not significantly associated with overall survival when it was measured in the tumor compartment, but it was associated, at least in the univariate analysis, when it was measured in the CD68 compartment. It is true that this significance didn't stand after controlling for multiple comparisons. This is perhaps a limitation of the study or a limitation of the cohort, because it's not perhaps well powered to to show significance for PD-L1, which we know is a marker associated with response and overall survival in lung cancer. But actually, this macrophage PD-L1 predictive value, we did find the same thing in a parallel study that was conducted by another member of the lab. In here, she quantified, or this was a work aimed to quantify immune cell PD-L1 in different immune cell populations. And as you can see, macrophages were expressing PD-L1 a lot. Actually, 80% of the immune cell PD-L1 or the stromal PD-L1 was coming from the macrophage. And again, as we found with the DSP, the PD-L1 in the macrophages was predictive of outcome, but PD-L1 in tumor cells was not, kind of giving us some sort of validation as well of the DSP finding, although this was done in the same set of patients, but with a different technology. So proceeding with those findings that we think are more relevant, or at least more they deserve further validation. This is actually one of them. I'm showing here that those tumors with high levels of expression of CD127 in the tumor, specifically in the tumor compartment, was associated with resistance. This is a nice example of why spatial information is important, because as you can see here, when CD127 was measured in the tumor compartment, those with high levels, those patients with high levels of CD127 in the tumor cells were doing worse. But when CD127 was measured in the CD45 compartment, this had no association with outcome, again, showing the importance of measuring in a spatial context. And this was another relevant finding in our study, that CD56 and CD4, when measured both in the CD45 compartment, were predictors of all clinical outcomes. This is showing the distribution of the quantification of, in this case, CD56 and CD4. And these are the Kaplan-Meier curves for progression-free survival here and overall survival, in this case for CD56 and CD4. And as you can see, both of them are markers quite strongly associated with both progression-free survival and overall survival. So in order to give more confidence on these findings, we decided to prove this outcome association with another orthogonal quantitative method. And we decided to validate CD56 as a marker of NK cells, because this was a more novel finding to us. So we decided to assess CD56 expression using a different quantitative method, in this case, INFORM. And this is a method that measures or counts cells. So we developed a panel to detect NK cells using cytokeratin, CD3 and CD56 as the marker of natural killer cells. And we did INFORM analysis as follows. First, we did tissue segmentation, in this case, tumor and stroma. Then we did cell segmentation to identify individual cells. And then we phenotyped these cells to distinguish between CD56 positive tumor cells. As you know, some tumors can express, or some tumor cells can be positive for CD56, particularly if they have neuroendocrine features. So we decided, we wanted to exclude or to distinguish between these tumor cells and CD56 positive immune cells. And also between these CD56 immune cells, we wanted to count separately those that are negative for CD3, which are NK cells, and those that are double positive for CD3 and CD56, which are NKT cells. So I'm showing now some pictures in order to show you that actually we could identify all these cell phenotypes. This is an image acquired with the Vectra system, and as you can see, we are detecting here tumor cells in green, CD3 positive lymphocytes in red, and CD56 cells in white. And as you can see, there are actually NK cells here, which are cells here in white, but there are also some NKT cells which are not very abundant, but you can find them here in the stroma, which are double positive for both CD56 and CD3. And using this orthogonal method, actually, we found a very similar, or we came to the same conclusion, that the number of CD56 positive cells in the stroma were actually predictive for both progression-free survival and overall survival, somehow validating our DSP finding with a degree of clinical outcome very similar to what we observed using DSP as our discovery tool. Here is showing the distribution, and also showing here on the left-hand side that those patients with clinical benefit had actually the proportion of cells in the stroma was higher, as you can see here. So these are the conclusions of this discovery work. As you can see, the geomics DSP system is well suited for immunotherapy biomarker discovery, as shown in this work. And these are the main reasons why I think this is important. First, because it is quantitative, because it provides a spatial resolution and information about cell-to-cell interactions, and also because it offers a comprehensive interrogation of what's happening in the whole context of the tumor microenvironment. And the main conclusion of our work was that we identified a number of candidate immunotherapy predictors in a spatial context that, of course, deserve validation in larger and independent cohorts. Remember that we only had one cohort. These findings need to be considered discovery findings that deserve validation in independent patient data sets or independent cohorts, and this is actually something that is currently ongoing. And with that, I just wanted to acknowledge, all the lab while I was at Yale, particularly David Ream, who was an extraordinary leader and mentor, and thanks also to the rest of the team that was supporting us during this work. Thank you very much for your attention.
Thank you, Dr. Zugazagoitia, for a very interesting presentation. Can you please clarify how many ROIs were selected for each non-small cell lung cancer case that you analyzed in your study?
Yeah, so this work was entirely performed on tissue microarrays. So for that, we assessed 2 tissue core per non-small cell lung cancer case, and these cores that were included in the tissue microarrays, they have 0.6 millimeters diameter. So we kind of draw a region of interest in each of these cores in the TMA. So at the end, we assessed 2 ROIs of about 0.6 millimeters, which is about 0.28 millimeter square tissue area per non-small cell lung cancer case, 2 per case.
Thank you. Do you think that a CD3 restricted compartment would have increased the performance of some of the biomarkers you tested?
Yeah, actually, this is certainly a possibility because, as I showed you before in the analytical results, some of the markers were expressed at low levels, or at least the signal to background ratio of some markers was quite low. And we believe that this might relate to the fact that these are markers that are expressed as particularly by T cells. So perhaps instead of doing these big CD45 compartments, perhaps a more restricted CD3 compartment could have increased certainly the signal of some of these markers, such as LAG3, which we know is explicitly expressed by T cells. So perhaps CD3-related compartmentalization could have increased the signal to noise ratio on some of these markers and therefore increase their performance. Yeah, this is certainly a possibility.
Thank you. In the orthogonal validation of the CD56 as a candidate predictor using INFORM, did you generate a CD45 compartment as you have done in the geomics analysis? Thank you.
Yeah, so for the orthogonal validation, we actually didn't create a CD45 compartment. We just segmented the tissue in two compartments. In this case, it was the tumor compartment and the stroma. So with this tissue segmentation with INFORM, the stroma actually contains the CD45 compartment that was created with DSP. This was perhaps some kind of limitation in our orthogonal validation, but as we came up to a very similar conclusion, we kind of believe that actually most of the NK cells that we were counting in the stroma using INFORM are actually found or actually located in a CD45 restricted compartment, as we saw with the DSP. But yeah, we didn't create a third compartment with the orthogonal validation.
Thank you. What do you think is the main novelty in this biomarker discovery work? Meaning, what does this study add to other similar, immunotherapy biomarker discovery studies?
Yeah, so I think the main novelty of the study is that we used a different approach to discover biomarkers. And this approach was that we were actually trying to find markers measured in a compartment-specific manner. We were trying to find markers associated with outcome in a spatial context. And I think this is the main novelty of this discovery study, that we actually demonstrate that it is important to measure markers, but also to measure them in a particular compartment, because this is where they are doing their work, and this is where the predictive capacity of these markers relies. And that's why this kind of technology enabled us to do this. And actually, we did find some really interesting discoveries in a spatial context that deserve further validation. But I think this is the main novelty, that we measure markers from different compartments in space.
Thank you. Finally, can you please comment about the clinical implication of the finding in these studies?
Well, perhaps it's a bit early to answer that question, because, well, as you know, this Geomics DSP is a research platform, so it's a bit early to talk about its potential clinical implications. Also, we need to consider that this was work done on unselected patients that received PD-1 or PD-L1 monotherapy, and we are kind of giving these drugs less frequently in unselected patients. But apart from that, I think that this kind of demonstrates or suggests that it is important to measure markers from specific compartments. And the DSP workflow is relatively simple, and it uses FFPE tissue samples. And perhaps, although it's a bit early to talk about this right now, but with further validation, this is an assay or a platform that could eventually make it into the clinic if these findings are confirmed in independent cohorts and larger cohorts. So just to give a brief summary of this work, and my take-home message would be that in this work we show the potential of the Geomics DSP platform to discover biomarkers of response in a novel way, And this novel way is related to this compartment-specific measurement of biomarkers of response. And this illustrates the potential of the technology, and these findings will need to be validated in dependent cohorts, but I think it's a very good start. These are, some of them are novel and interesting findings that we will actually try to validate in future cohorts. Thank you very much.
Thank you again, Dr. Zugazagoitia, for your presentation and discussion.